Good morning, students. Welcome to today's lesson on methods of teaching biology. In this lesson, we are going to learn about organizing laboratory-based practical lesson. And this is the last session of Unit 4. So by the end of this session, you will be able to manage the biology laboratory and then also organize biology practical lesson. Now, before we start, biology is a subject that needs to be taught with more emphasis on practical work because biology is more of the environment. So we are saying that when teachers teach only the theory of the content, students find it difficult to understand the concept. And they also lose out on essential scientific inquiry skills. So if the teacher is going to teach practical work, then it is important that he knows how to organize such a lesson. So now we are going to know the ways of managing the laboratory. First one is keep an inventory of all the equipment and consumables in the laboratory. So when you have a lab, when you have a biology lab, you have to keep inventory. Here, what we are trying to say is that you have to know the consumables available. If some is available or if some is not available, you have to know. Or if in stock or out of stock, you have to know. So the University of Cape Coast has procured consumables for our study centers. That is the science and maths education, they have 10 study centers. So if you are doing biology, chemistry, physics, your study center, there is a laboratory there. So the University of Cape Coast have procured consumables or have stocked the labs with specimen or laboratory equipment. So we are ensuring that what you are going to use for the practicals are available. Now the next point is make sure all equipment are good working conditions. Yes, before you even conduct a practical, you have to ensure your equipment are working. The next point is keep an inventory of all breakages that occur in the laboratory. Yes, in some lab, we say breakages are payable. So students have to be careful when they go into the laboratory. If your lab instructor has not informed you to pick any apparatus, you do not pick. You follow instructions in your lab. Now the next one is have a minimum stock of consumables at which you will not exceed. So when the consumables reach the level, then you have to replenish them. That is very important. You have to know the minimum stock of consumables you have to use in the laboratory. So when that minimum is or when that stock is finished, then you have to replenish it. The next point is prepare rules and regulations for students' conduct. So that is the behavior of students. Students in the lab, they just go here, come there, pick any item in the lab. Some of them are very corrosive. They are not aware. They just pick. They are new in the environment, so they want to have some skills, manipulative skills, how to use some equipment in the lab. So as a teacher, you have to prepare rules and regulations for students' conduct. And we are saying that such rules should be made visible to students and anyone who enters the laboratory. So most of the times I advise that when you make the rules, you sellotape it on the table, on the bench, laboratory bench, or you paste it on the table or you paste it on the wall. As soon as you enter the lab, you see that do not eat in the lab, do not drink in the lab, do not spill water in the lab, do not throw items in the lab. A whole lot of rules that will help you to maintain your lab or manage your lab. The next point is students should be instructed on how to care for apparatus and instruments. Yeah, you have to know, you have to instruct students, like tell them, do not touch this, touch this. You tell them how to handle laboratory equipment. Now, we are moving to steps in organizing biology practical session. So the First step, you're saying that if students are to enjoy practical lessons and be mentally active, then the teacher should be proactive in organizing practical lessons. So the following steps can help teachers to successfully organize biology practical lessons. And the first step is all preparatory work should be complete before the practical lesson begins. That is advanced preparation. You, the teacher, you have to prepare in advance to know what to do before the practical is commenced. The next step is teachers should ensure that all equipment, specimen, reagents, and anything to be used are in good working condition before the experiment. So you have to check whether the equipment you are going to use to conduct the practical are in good shape or are in good condition before you commence the practical. Teachers should perform the experiment and make sure that the right outcome is achieved before students come to the laboratory. So as a teacher, what you expect students to know, you have to prepare ahead to know that okay, everything is in good shape, everything is in good condition. This is the results I expect. And therefore when students come, I expect they have to see this result or they have to get this result and present. The next point is students should know beforehand what work they are to do and should come to the class prepared with appropriate instruments. So you inform students that this is what we are going to do. Next week when we meet, we are going to do practicals on dissection. So this is what you'll be expecting, this is what we are supposed to do. You are supposed to read this book. You give them some reference materials to go and read on before they come. So they have knowledge about what they are coming to do. So we are saying that if instruments will be provided by the teacher, then they should be ready by the time students come to the laboratory. Yeah, that is. The next point is instructional materials such as specimen, reagents, slides, and worksheets should be ready for immediate use. Yeah. So all these materials should be ready for the practical session. Make sure all specimens are there, reagents are there, slides are there, worksheets are there for students to use. The next point is the teacher should demonstrate what is to be done to students if need be. So you do some demonstration for students to see, and then after that you leave the work for students to do, and then you supervise. The next point is if it is guided entry, steps to be followed should be ready on each workstation or made available to students. Yes. So if you are giving some guidelines or a step for students to follow, then at every station you provide the steps for the students to follow in order to conduct the practical. The teacher should be aware of potential dangers of the practical lesson being conducted. So you, the teacher, you have to know of the dangers. So that's why there should be safety precautions for students to know in order not to cause any harm. The next point is appropriate precautionary measures should be made available for students. Yes, that is very important. And we're also saying that to ensure that students abide by the laboratory rules at any point in time. You have to ensure that students go by the rules you provided when they entered the laboratory. We've learned a lot in this session, and then we've ended the session, so we have to review what we've learned in this session. So, in reviewing what we've learned, I want you students to answer this question. One, mention two ways of managing the biology laboratory. Two, mention the steps in organizing a biology practical session. And I believe when you find answers to this question, it means you have understood or you understood the session.